In Chapter 22, we're talking about nucleic acids. We'll start out by talking about what makes a nucleic acid and being able to identify the differences between DNA and RNA. We'll then move on to talking about primary and secondary structure in nucleic acids, and then we'll talk about how DNA is replicated and how DNA is eventually translated into protein. We'll also talk about things that can go wrong with DNA, like mutations and viruses. Our fourth type of biomolecule is a nucleic acid. Nucleic acids include DNA and RNA, and even though nucleic acids make up a very small percentage of our overall biomass, they play an outsized role in the function of our cells because they contain all of the genetic information that is eventually translated into proteins, the largest bioorganic component of our cells. DNA is a type of nucleic acid. A nucleic acid would be any polymer whose units are nucleotides. Of course, nucleotides then need to be defined as well. Those are any molecule that's made up of ribose, which is a carbohydrate, phosphate, and some sort of nitrogen heterocycle, a ring that contains nitrogen. We can see those pieces at play in our different nucleotides. On the top, highlighted in purple, you see the different types of bases that are present. Remember, we had talked about the structures pyrimidine and purine in Chapter 17 as being particularly important because those are the structures that were present in DNA. So you can see the top row in purple are all pyrimidine bases. They all have the 2 nitrogens in the aromatic ring. And the bottom 2 are adenine and guanine, which are purine bases. They have the 4 nitrogens in the 2-ring heterocycle. Those bases will all be connected through something very similar to an acetal bond to either a ribose or a deoxyribose. A ribose for RNA, a deoxyribose for DNA. And each of those will be connected to a phosphate or multiple phosphates through a phosphoester bond at the final alcohol position, at the 5 position. In both DNA and RNA, we have 4 bases that we use to encode all of the information that we use in the nucleic acid. However, the bases that we use for DNA and RNA are different. In both DNA and RNA, we use both purine bases, adenine and guanine. In both DNA and RNA, we also use cytosine, one of our pyrimidine bases. However, the other pyrimidine base is different depending on whether we're in DNA or in RNA. In DNA, we use thymine. In RNA, we use uracil to accomplish the same maintenance of information. Notice the difference between thymine and uracil is only the CH3. So other than that CH3, those bases are identical to each other. DNAs are really just a short way of saying deoxyribonucleic acid. Deoxy meaning that the ribose itself is deoxygenated, and ribonucleic acids would then be just the straight ribose for the carbohydrate. We can see this difference in the deoxygenation if we look at RNA and DNA side by side. If we look at the RNA structure, we'll see that the ribose that is used for RNA has an OH on the 2nd position. In DNA, that OH is replaced by a single H. That would be the deoxyribose that is used in DNA. Again, we see that thymine and uracil are different, while all the other bases are the same between DNA and RNA. We also will talk about the possible nucleotides a little bit different for RNA versus DNA. We will abbreviate them with a 3-letter code. The first letter code of the code refers to which base is used. The second letter, the M in this case, refers to how many phosphates are present, and the P in this refers to that there is phosphates. So for AMP, it stands for adenosine monophosphate. GMP would be guanosine monophosphate, etc. If we had ADP, it would be adenosine diphosphate, and ATP would be adenosine triphosphate. Notice that's a little bit different for our possible nucleotides for DNA. They always come with a D in front of them indicating deoxy. So then we have deoxyadenosine monophosphate or deoxyguanosine monophosphate, and so on. While certain nucleotides, like adenosine triphosphate, are relatively ubiquitous throughout the cells, many of our nucleic acids actually come as polymers. Each of those polymers are going to be linked by the sugar, by either the ribose or the deoxyribose. So the basic structure of any nucleic acid is going to be phosphate linked to a sugar, which is linked to the next phosphate, which is linked to the next sugar, which is linked to the next phosphate, and so on. So for DNA, it would be phosphate linked to deoxyribose, since that's the only sugar, to a phosphate, to deoxyribose, and so on. Whereas in RNA, it would be phosphate linked to ribose, linked to phosphate, linked to ribose, and so on, since ribose is the sugar that's used. Notice that in either of these cases, the base that is used is not part of the backbone. It's always a side chain, much like the side chains of amino acids are not part of the backbone. The primary structure of the DNA and RNA is going to be linked together through those phosphoester bonds. We also talk about these nucleic acids with a distinct directionality. We always list them from the 5' end to the 3' end. That's because that's the direction that they are synthesized in our body. The 5' end would be the end that has the 5' open. Notice that the 3' is where it's linked to the next carbohydrate in that 5' end. And the 3' end of the entire nucleic acid would be where we have an open alcohol on the 3' position. When we link all these together, the phosphates are always going to be connected as diesters, and they always carry a -1 charge, which makes nucleic acids really quite anionic. They're really quite water soluble. The backbone is always going to be the same in every one of these cases. Just like we talk about the sequence of amino acids as the primary structure of a protein, we also talk about the sequence of our nucleotides as the primary structure of our nucleic acids. Our DNA and RNA also have secondary structure on top of the primary structure. RNA secondary structure is widely varied and relatively difficult to understand. It's outside the bounds of this class in most cases. DNA secondary structure, on the other hand, is relatively straightforward and important for how our bodies maintain consistent information across many generations in our genes. DNA will automatically arrange into a double helix, which is shown on the left. And in the double helix, 2 strands of DNA will be held together by hydrogen bonds between complementary strands. Notice in the structure on the very left, there's a strand that it goes from top to bottom, starting at 5' and ending in 3'. The complementary strand goes in the opposite direction, starting at 5' on the bottom and going to 3' at the top. For this reason, we talk about DNA complementary strands as reverse complements. They're going in opposite direction and they are complementing each other. When we say the strands complement each other, we mean that every time we have a certain base pair, it always has a pair that goes with it. Those pairs are determined based on 2 different factors. In order for the DNA double helix to maintain the same distance between the helix at any given point, we always have to have the same size base pairs. So we'll always have exactly 1 purine and 1 pyrimidine, so that they take up the same amount of space. We could have 2 different forms of T, in this case, pairing with a purine. So here we have T, which is a pyrimidine, pairing with A, which is a purine. And here we have T, which is a pyrimidine, pairing with G, which is a purine. Notice that when T pairs with A, there are 2 hydrogen bonds that connect these 2 bases. That's a much stronger interaction, and so T will always match up with A. We say that T and A are complementary. Similarly, C, which is a pyrimidine, can either match up with G or with A. When C matches up with G, we end up with 3 hydrogen bonds between those, whereas when we match C up with A, there's only 1 hydrogen bond. The C and G matchup is much, much stronger, and because of that, we end up with C always being the complement for G. Knowing what we do about DNA complementary strands, If one strand is given, we can always know the other strand is in the reverse direction and complementary. Take a look at the first strand shown and try to identify what the reverse complementary strand would look like. The second strand would go from the 3' to the 5' direction. Reading from the 3' direction, there would be an A underneath the T, a T underneath the A, and A, T, C, G, A, T. So for every T is paired with an A, and every C would be paired with a G, but the strands would be in opposite direction. Now pause and try to identify what the second strand would look like. Again, we see every C would be paired with a G and every T with an A, but again, the 3' end would match with the 5' end and the 5' end with the 3' end. The strand would be in the opposite direction. Because our base pairs have exact matches, these strands can always be separated and we can synthesize the reverse complementary strand and create new DNA. The matching always is going to take place from the 5' end to the 3' end by an enzyme called DNA polymerase, which matches the direction that we write our DNA in. Notice that if we're trying to create DNA from 5' to 3', that works really well when we start at the 3' end and work the opposite way. It doesn't work as well in the complementary strand, and so the complementary strand is created in piecemeal segments called Okazaki fragments that are later attached together. It's pretty difficult to separate all the hydrogen bonds that hold DNA together, so it happens just a little bit at first, and as complementary strands are synthesized, it becomes easier and easier to pull apart the DNA as it replicates. We call the places where the DNA starts to pull apart origins of replication, and eventually, as the origins of replication spread, we end up creating 2 strands of DNA from the single template. This whole process is called DNA replication, and it creates 2 exact copies of DNA from the one original DNA molecule. These DNA strands make up our chromosomes, which are complexes of the DNA and protein that together store our genetic information.